My name is Claudia Gaona, and today we're going to go over aspartame and saccharin and the information about their safe use in the food industry. To start off, saccharin and aspartame are high-intensity sweeteners used as sugar substitutes or sugar alternatives. These substances are sweeter than sugar but contain fewer to no calories. Both aspartame and saccharin are high-intensity sweeteners but have slightly different characteristics and are used in different sugar brands. Saccharin is used in Sweet and Low, Sweet Twin, and NectaSweet. Aspartame is used in NutraSweet and Equal. When individuals think of high-intensity sweeteners, they recognize these brands rather than the actual names of aspartame or saccharin. Saccharin is 200 to 700 times sweeter than table sugar, does not contain calories, is approved for use as a non-nutritive sweetener, and is approved for use in certain beverages, fruit juice drinks, and mixes. On the other hand, aspartame is 200 times sweeter than table sugar, does not contain calories, is approved for use as a nutritive sweetener, and has been approved for use in some chewing gum, dry bases for foods like beverages, instant coffee, tea, gelatins, and puddings, as well as dairy products. Saccharin was first discovered and approved for use in 1879. In the 1970s, it was linked to the development of bladder cancer in laboratory rats, and because of that, saccharin-containing products had to have a warning label letting consumers know of possible carcinogenic effects. Then in 1981, it was placed on the national list of potential carcinogens, and it was not until 2000 that it was removed from that list by the National Toxicology Program of the National Institutes of Health. In the year 2000, the label mandate on saccharin-containing products was removed due to study findings on its safety. On the other hand, aspartame was approved for use as a tabletop sweetener in 1981. Then in 1983, the FDA approved its use in carbonated beverages and syrup bases. In 1996, the FDA approved its use as a general-purpose sweetener, and in 2005, a study was published that raised the possibility that high doses of aspartame cause lymphoma and leukemia in laboratory rats. A follow-up study was conducted after that 2005 study, and it was concluded that high intake of aspartame-containing beverages was not associated with lymphoma, leukemia, or brain cancer. Then, a 2013 study found no association between aspartame and cancer risks. Some of the main concerns with saccharin since its approval was the possibility that it could be linked to the development of bladder cancer in laboratory rats in the 1970s. However, after that study was published, there were more than 30 human follow-up studies, and they found that the lab rat study findings were not relevant to humans. This was mostly because the urinary bladder cancer in lab rats was related to the physiology of the rat's urinary system. Therefore, the results were not the same in humans. Concerns over the association between saccharin and urinary bladder cancers in diabetic patients also arose. However, follow-up epidemiological studies and case control studies have been conducted, and they all showed no clear association between saccharin intake and urinary bladder cancer. One of these studies was the Armstrong and Dawes study that examined time trend data for urinary bladder cancer in humans. The data from this study showed no clear indication that increased use of saccharin or artificial sweeteners starting in the 1940s was associated with a general increase in urinary bladder cancer when confounding factors like smoking were controlled for. The risk for urinary bladder cancer in diabetics, who presumably consume greater amounts of artificial sweeteners than the general public, were found to be at no greater risk than the general population in this study. A group of case control studies from 1980, 1981, 1982, and 1983 were also conducted, and they all looked at the associations between the use of artificial sweeteners and urinary bladder cancer in diabetics, to which they found no statistically significant association. One of the main concerns with aspartame was due to a 2005 study conducted by the European Ramazzini Foundation that claimed high doses of aspartame were linked to lymphoma and leukemia in laboratory rats. But after the study results were published, the FDA asked to review the entire study. but the researchers only provided the FDA partial study data, and even then, the FDA was able to identify shortcomings in the design, reporting, and interpretation of the study. After that, the FDA concluded that aspartame was safe and those study results were invalid. Concerns over aspartame intake and brain cancer have also emerged, but a 2006 study by the National Cancer Institute on retired individuals found no association between aspartame intake and cancer, lymphoma, or leukemia. A 2013 epidemiological data review study was also able to show the lack of association between aspartame intake and cancer. Other health concerns that have emerged over the years have been the consumption of saccharin and aspartame and their possible effects on glucose intolerance, decrease in energy balance in humans, the possible links between consumption and insulin resistance, consumption effects on body weight and obesity, energy intake, appetite, and the effects they can have in the inability of individuals to control quantities. These concerns have emerged over the years, but due to the lack of exposure data and limitations of available epidemiological studies, the response to those concerns has not been properly addressed. Long-term studies that look at consumption of these sweeteners, the amounts, and the possible health effects they have later on in an individual's life are needed, but that takes time. However, as of now, researchers along with the FDA have deemed these substances as safe. In terms of exposure, humans are exposed to these two substances through oral consumption of foods or beverages. The dose-response relationship has not been established to reflect a precise amount, but high rates of consumption can possibly affect those with chronic diseases or those who overuse them. The most susceptible populations at risk of suffering negative health consequences due to the intake of these substances are consumers with chronic illnesses who do not limit their sugar intake, individuals who opt for food or beverages that contain these sweeteners, thinking they are a healthier choice, or those who overuse them. Some of the solutions to these concerns, besides the current efforts, include new risk management interventions where educational trainings or workshops are conducted in an effort to increase awareness on what these sweeteners are and the differences between them and regular table sugar. In these types of interventions, the idea that moderation is key needs to be one of the main messages. Further long-term epidemiological studies and other scientific studies that focus on priority concerns are also needed, especially long-term ones that look at current intake and future consequences for high intake of those sweeteners. These types of studies can be very lengthy, but the results can be very valuable and help us better understand and manage the consumption of these substances. Changes in label policies are also needed. Manufacturers should advise consumers the importance of moderation being key, and the possible side effects of high intake of these substances can occur. Based on the scientific evidence from some of the studies mentioned and through the review of the literature, aspartame and saccharin are not toxic and therefore safe to use, but with moderation.